Well, good morning, everyone, and welcome to the um, ICEDA Averta Awards webinar. My name is Glenn Vanderkraak, and I'm pleased to host this webinar. And just as a reminder, ICEDA is a research network that is Canadian-based, and it's focused on advancing knowledge on endocrine disruptors and supporting research excellence. Today's webinar is going to highlight some of the work of our early career researchers and then also some of our established researchers. And we've got a series of 4 webinars that will be delivered this morning, and I'm very much looking forward to hearing those presentations. We are very grateful to have partners that have shared our commitment to this research topic area and to support the next generation of scientists. And with that, it's my pleasure to introduce Delphine Delonre, who represents the Averta Foundation, which is a proud supporter of the ICEDA Awards. Delphine, would you like to say a couple of comments, please?
I don't see Delphine. I think she had trouble to join,
Oh,
but maybe she can join at another time today. So maybe we go on.
okay. Um, thanks. Well, um, sorry, it'll just be a second. Would you like to move right to the scientific program, or would you like to make a few comments, Romy, about the setup for today?
Yeah, some comments here. Good morning, everyone. Again, my name is Romy. I'll be facilitating the webinar today. And just to remind you that this session is recording, and the recording will be shared afterward on our YouTube channel. All participants are now muted, and if you have any question, please submit them by the Q&A. You know, you have a symbol with a question mark and Q&A at the bottom of your screen. And please use the chat only if you have technical issues. After each presentation, we will have 5 minutes of question. I will read it out loud, and we will answer as many possible questions. So you will have to after each presentation. Thank you.
Well, thanks. And maybe we can get started
Yes.
on the scientific program. And our first speaker today is a master's student at the University of Ottawa, and she's Lai Wei. And she was the recipient of the 2025 CIAPE Everta Masters Award. And her work focuses on the development of new approach methodologies using zebrafish models to better understand the effects of environmental contaminants. And as listed on this slide, she's going to talk about the development of a zebrafish new approach methodology for high throughput screening of PFAS-induced beta cell disruption. Lai, please go ahead with your seminar.
Thank you so much, Glenn, for the introduction. I'm just going to share my screen. Okay. Well, good morning, everyone. My name is Lai, and I am a master's student that is co-supervised by both Dr. Yan Menigan and Dr. Jenny Bruin from both University of Ottawa and Carleton University. My project focuses on using zebrafish they're called Daniel Rarios, as a new approach methodology for high throughput screening for PFAS effects on pancreatic beta cell development, specifically in type 2 diabetes. Well, the animal model that I am working with, of course, is called zebrafish. And many of you may not be familiar with this model, so I will just briefly introduce the advantage of using the zebrafish model. Well, to begin with, due to its size, they require a very small housing space, and which makes them extremely cost efficient. They have the ability to produce all year round with externally fertilized translucent embryos that allows for live imaging as well as fluorescent microscopy. They undergo rapid organogenesis and they reaches sexual maturity in about 3 months. This is very crucial for early development toxicity testing because we're able to work with fully formed organs during our exposure. And in this case, one female zebrafish can lay up to 300 eggs in one night, and that permits the high throughput screening aspect of chemicals. And they also share a 70% gene similarity with humans, which makes them an excellent model for toxicity testing. And one important thing to note is that they're also a ethically acceptable model that allows for the 3 Rs, which stands for refinement, reduction, and replacement. Under the new EU directive that is circulated around the animals in scientific purposes, the earliest life stages of zebrafish is not classified as protected, which means that they actually do not fall within the regulatory frameworks governing animal research. Exogenous feeding for zebrafish is considered the stage where free swimming larvae is subjected to animal protections. And zebrafish only starts exogenous feeding at 5 days post-fertilization, which makes them exempt from animal regulatory guidelines during early life development and an excellent model for toxicity testing. And diabetes is considered one of the fastest rising global chronic metabolic diseases, with cases doubling in the last 30 years to over 800 million adults worldwide. And in Canada, there are around 2.9 million Canadians that are currently diagnosed with one type of diabetes. And out of these 2.9 million people, 90% of them is diagnosed with type 2 diabetes. The pathogenesis of type 2 diabetes is manifested by two different factors. The first one is a beta cell dysfunction, which is caused by an impaired glucose-stimulated insulin secretion. And the second one is insulin resistance, which is caused by a loss of a peripheral glucose uptake. And it's the combination of these two factors that leads to progressive beta cell loss and results in type 2 diabetes. Type 2 diabetes develops through multiple interacting pathways. While we only usually focus on the traditional risk factors, that includes lifestyle choices like diet, physical activity, age, and genetic dispositions. These alone don't really fully explain the rapid rise in diabetes prevalence, particularly in developed nations. And over the past decade, researchers have increasingly recognized that environmental factors and exposures play a critical role, but often overlooked in metabolic dysregulation and diabetes development. And out of these contaminants that is being focused on right now, one important class of environmental pollutants that is gaining attention is called PFAS. And this is the focus of my project. PFAS stands for poly and perfluoroalkyl substance. They are not the greatest chemical. Well, why is that? It's because according to different human epidemiological and animal manipulation studies, some of these chemicals are suspected of being called endocrine disrupting chemicals. And that includes this large class of PFAS. So PFAS is man-made compounds that is currently active in 6,000 different compounds that is made out of carbon, fluorine, and other elements. These compounds combine to make an alkyl chain that is typically 2 to 16 carbon atoms in length. And they have different functional groups, charges, and chain length, and they contribute to their persistence and different toxicity. These chemicals are often referred to as forever chemicals because of their resistance to degradation, their potential carcinogenicity, their bioaccumulation effects, as well as global distribution across the world. They are widely discussed as an endocrine disrupting chemical because of their evidence that is linked to altering thyroid signaling, steroidogenesis, and reproductive or metabolic outcomes. Although the exact mechanisms of these PFAS compounds are not fully resolved, the key to understand their environmental fate is their surface active behavior. Well, their fluorinated backbones is both hydrophobic and hydrophilic. That means their functional head groups are hydrophilic, which allows them to partition between intersurfaces, typically between water and air. And as a result, it makes them highly stable and extremely persistent to any type of degradation that contributes to their persistence in the environment as well as human blood. There are two major PFAS chemicals that are referred to as legacy PFAS, and that is PFOA, PFOA, and PFOS. They are already banned in Canada currently, while they are currently restricted under different Canadian environmental protection acts. There are many newer PFASs that have actually entered the market to serve as replacements for these legacy PFASs. Because of the EDC aspect of these metabolic disrupting effects of PFAS, we are concerned that it may contribute to pathways that is relevant to type 2 diabetes pathogenesis. And that is also the focus of my project, which is what is the link of PFAS to type 2 diabetes? Well, currently, many studies have already begun to explore the relationship between PFAS and type 2 diabetes. And several studies have already shown a positive correlation between type 2 diabetes and different glycemic indicators from the exposure of PFAS. The first one focused on the plasma PFAS mixtures that is actually positively associated with a 31% increase of type 2 diabetes incidence. The second paper talked about the maternal plasma PFAS concentrations that is measured during pregnancy were also positively associated with plasma insulin levels even years after delivery of a child. And the third longitudinal study shows that PFAS worsens insulin resistance and beta cell function in women with prior gestational diabetes. But as I mentioned, there are many PFAS that came into the market to serve as replacements for the legacy PFASs. How do we know which ones actually affect type 2 diabetes risk? Well, that is where my project comes in. I have used the advantage of a zebrafish and their fully sequenced genome, and I made a transgenic line that is called the INS-M cardinal line. So essentially, on the slide on the left side, it shows the plasmid map. So this plasmid was initially injected into the zebrafish embryo at 1 cell stage through a total transgenesis cold fusion. And this plasmid actually fluoresces their beta cells. So as you can see, from 3 days post-fertilization to 5 days post-fertilization, this M-cardinal protein fluoresces the beta cell mass in the zebrafish larvae, which allows me to visualize their beta cells in pancreas for zebrafish. And this becomes very useful because I am able to expose a zebrafish larvae in a span of 5 days and to see the effects of the chemical on developing zebrafish, especially in the pancreas, just by gauging the size and the mass intensity for the fluorescent protein that is tagging the beta cells. Well, the first thing I would like to focus on is at the end of the exposure for my experiment, the EC50 value. So for those who are not familiar with a EC50, EC50 stands for half maximal effective concentration. This is a key measure in pharmacology that represents the concentration of a chemical that is needed to produce 50% of its maximum possible response in a biological system. Well, in zebrafish, we usually gauge it through a series of morphometric analysis and lethality as shown in panel A. So from these results, we're able to develop something called a dose response curve that is shown in panel B. This is a sigmoidal curve that is widely used to calculate the EC50 value of the desired compound, pointing right here. Using the methodology that I have just mentioned in the previous slide, I have tested 11 different PFAS compounds so far. We are already seeing a trend for the different effects based on different chain lengths. The longer chain PFASs appear to be more toxic than the shorter chain PFAS. And these compounds are selected from Health Canada prioritized PFAS lists. And as well, there are 7 different PFASs that is pointed with an arrow. These compounds are listed from the donor tissues that are processed from the Alberta islet core, and from them, they obtained plasma samples from donor tissues that were sent to a company called SGS Access, in which they did a mass spectrometry analysis to measure a panel of 40 different PFAS compounds in each of the donor plasma and pancreas. And this is a work of one of the PhD candidates in the Bruin lab, and he has narrowed the list to these 7 different PFAS, and they were detected in over 70% of the measured donor population. And my next steps will to look at the specific 7 different PFASs that are detected in more than 70% of the donor population, and to gauge the effects of PFAS on developing beta cells in zebrafish larvae. Well, as I mentioned, towards the end of the experiment, I have obtained all the EC50 value for different PFAS chemicals. I will perform the same exposure experiment, but with a sublethal doses. So this means that EC50 value actually becomes my highest dose and is followed by half logs down the concentration for a total of 5 different doses, along with a DMSO vehicle control. And the endpoints that will be tested is non-invasive live imaging through the fluorescently tagged pancreatic beta cells, as well as gene expression. For today, I will only briefly go over the preliminary results for some sublethal exposures of PFAS, which is a legacy PFAS chemical that is already banned in Canada. For gene expression, I ran 4 different glucose-responsive genes, and from the sublethal dose of PFAS, we saw a significant decrease in INS-A expression levels. And INS-A, what's so special about INS-A in especially zebrafish is that it encodes for pre-proinsulin. And this gene processes into mature insulin hormone that is specifically found in the pancreatic beta cells. So disruption of this INS-A gene disrupts the larval glucose homeostasis. And with that, I would like to thank all of my lab members, as well as Dr. Menigan and Danny Bruin for the support. And I would like to open the floor for any questions. Thank you so much.
Well, thank you so much, Lai. I quite enjoyed your presentation. Do we have any questions from the audience?
No, we don't have yet.
Okay. Um, I have a quick question for you. When you first calculated the EC50, you based your analysis on what you measured on 5 days. Did you ever do time course experiments to find out when you could detect a response? And did this relate to the turn on of the pancreatic cells? And when was that first noticed?
That's a it's a very good question because before I started doing the long 5-day chemical exposures, I was trying to gauge at when the chemical was actually affecting the beta cells. So I
Yes.
started with um 2 hours of chemical exposure, 2 hours, 4 hours, 6 hours, 12 hours, and 24 hours. And from all the papers that I read, is that the pre-transcription factors and some of the early on genes that gets turned on to make beta cells in zebrafish gets activated at around 10 to 12 hours post-fertilization. And that is also why I decided to start the exposure at 12 hours post-fertilization to ensure that during the early on development of even the beta cell precursor cells, I can have the PFAS effects on these beta cells. And as for the time course for the experiment, um, because we wanted to do full 5-day exposure, we didn't really stop at any specific day, but I did track mortality and lethality every single day just to see, because I was also curious on when exactly things started to really accelerate. And it's usually around 3 to 4 days. And that is actually when zebrafish they shed their chorion, so their egg shell. So that's
Yep.
when the effect actually started to kicking in, if that answers your question at all.
Thank you.
We have another question from Isabel. Mechanistically, do you have any hypothesis why long chains are more toxic?
Um, from my understanding, because like I talked about in the beginning, the characteristic of PFAS is that they are extremely persistent to degradation because of their long carbon fluorine chain that is very hydrophobic. So when they have longer chain, it makes them more hydrophobic and it makes them more persistent compared to the shorter chains because with shorter chains, we can kind of break it down easy. Well, not, it's all relative, but it's relatively easier to get broken down compared to the longer chains. And I think that's one of the reasons why it's more toxic in zebrafish because they're less likely to metabolize the longer chain PFAS.
Hmm. Well, Lee, thank you very much for your presentation. I did really enjoy that. But I think we'll move on to the second presentation.